Good morning, everyone. The energy in this room is just, it's amazing. If you could please take your seats, we can start the Proctor Award ceremony. My name is Goldis Malik. I'm the Retinal Cell Biology Trustee, and I'm happy to join you today to review the history of the Proctor Medal Award. This medal was established in 1949 as a memorial to Dr. Francis I. Proctor, an ophthalmologist who conducted extensive research on the etiology and treatment of trachoma. This award honors outstanding research in the basic or clinical sciences as applied to ophthalmology. The 2025 Proctor Medal Award is presented to Christine Curcio, PhD, Professor Emerita of Ophthalmology and Visual Sciences at University of Alabama at Birmingham, and Cynthia Owsley, PhD, the Nathan E. Miles Endowed Chair of Ophthalmology, also at University of Alabama at Birmingham. In their individual research careers and as long-term time collaborators, Dr. Curcio and Owsley have made seminal research discoveries in the field of age-related macular degeneration, enabling the development of targeted treatment and prevention strategies. I'm pleased to present this year's Proctor Medal to Dr. Curcio and Dr. Owsley. If you would both join me on the stage, and please come forward. Next, I'd like to introduce Dr. Maximilian Pfau, Senior Principal Medical Director at Roche and Head of the Visual Neurophysiology Platform at the Institute of Molecular and Clinical Ophthalmology in Basel. Dr. Pfau will review more of Dr. Curcio and Dr. Owsley's achievements and introduce their lecture.
Good morning, everyone. It is my great pleasure and honor to introduce you today to our 2025 Proctor lecturers, Dr. Cynthia Owsley and Dr. Christine Curcio. Cynthia Owsley earned her undergraduate at Wheaton College, and subsequently she pursued her PhD in experimental psychology at Cornell. Then she went on for postdoctoral training at Northwestern, where she started specializing in visual psychophysics, a field that she would go on to transform profoundly. In 1982, she joined as faculty at the University of Alabama, Birmingham. Christine Curcio started her scientific career at Brown University, can recognize her early on already, and she subsequently pursued her PhD at the University of Rochester, where she specialized on neurobiology and aging, and then went on for postdoctoral training in Boston and at the University of Washington in Seattle. There she joined Anita Hendrickson Labs, focusing on the histology of the retina, a field that she would profoundly transform in the next 3 decades. And in 1990, she joined as faculty at UAB. For me, it's really important to emphasize their impact, and there are 3 facets of the impact to their work. Of course, there's this very traditional academic or bibliometric impact, which is immensely impressive for both of them. Cynthia has an H index of 89 as of yesterday, with more than 29,000 citations. Christine has an H index of 94 with more than 37,000 citations. But in my mind, what matters much more is actually the real-world clinical impact they had, and then also how they did research and the broader scientific impact. In terms of Cynthia's hallmark contributions, she was the first one to really demonstrate in large, well-powered studies that rod photoreceptor dysfunction precedes cone dysfunction, age-related macular degeneration. In that context, she also invented a new format of clinical studies. Beforehand, there were small case series in the field of IRDs with advanced psychophysical testing, and then large-scale studies with mostly just fundus photography imaging and BCVA. And she was the first one to merge those 2 concepts and come up with large, well-powered studies that included advanced psychophysical testing. And she was also the first one to demonstrate that slowing of the rod-mediated dark adaptation is predictive of future age-related macular degeneration. And in terms of real-world clinical impact, she was involved in the early development and validation of the ADAPT-DX device, a dark adaptometry device now used at clinical sites across the world. And also, she highlighted the value of full contrast sensitivity function testing, a vision test that is gaining more and more traction in the field of AMD currently. Christine's contributions are equally impressive. She contributed the photoreceptor topography maps, which are one of the most cited publications in ophthalmology overall. She elucidated the mechanism of drusen formation, the role of lipids and lipoproteins, and she established the histologic correlate of most OCT findings in neovascular AMD. And what does it mean with real-world clinical impact? Her photoreceptor maps are actually basically published in every physiology textbook across the world. Every medical student has seen them at some point. There are now several pharmaceutical companies focusing on lipid-targeting therapies in AMD. And also her OCT correlation studies contribute to anti-VEGF clinics on a daily basis to answer when to inject and maybe when not to inject, for example, in the case of ultraretinal tubulations. And what's even more impressive to me as a young scientist is not only what they did, but how they did it. So very early on, they showed a huge commitment to open science. A long time before the term open science even existed, they pre-specified and pre-published their hypotheses. They also shared raw data of their studies a long time before there was any NIH mandate to do so. They participated in open peer review. Sometimes I liked their review comments, sometimes I didn't like them, but they definitely helped to improve manuscripts. And they mentored numerous students, both within their lab, but also they helped people outside of their lab significantly. So really, thank you for that. And of course, science is teamwork, and they built up an impressive research team with long-term collaborators. Here on the left, you see Cynthia's psychophysical clinical research unit, also with Gerald McEwen, a long-term collaborator of her. And on the right, you see the lab of Christine Curcio, and also some long-term collaborators like Rick Spade from New York or Bailey Freund. And also takes a team at home too. And funnily enough, both of them are married to a scientist named Dr. Sloan. Here you see Cynthia with her husband, Mike Sloan, a renowned psychologist from Ireland, who also held a faculty position from 1982 onwards at UAB and also jointly published with her. And here you see the other Dr. Sloan, Dr. Kenneth Sloan, who is a renowned computer scientist with a PhD from University of Pennsylvania, faculty positions at MIT, Washington University, and eventually also at UAB. And he also published jointly with Christine a lot. And they, of course, have both a lovely family and were also lovely mothers to their children. Here you see Cynthia with her two children, Peter Aiden and Maeve Mary. And here you can see Peter now with his wife, Catherine Shelton. Both of them are MDs. He's currently in anesthesiology residency. She's a trauma surgeon. And here you see the daughter, Maeve, with her rough collie, Banjo. Here you see Christine's family. Again, she has two sons. One of the sons is also called Peter, another parallel in their life. So she has two sons, David, who works as a computer scientist at Google, and Peter, who is currently pursuing a PhD in music, and they have two lovely grandchildren and also jointly do music. And here you can see Ellie and Sam, the grandchildren. So please join me in congratulating and welcoming our 2025 Proctor lecturers. Congratulations again. I'll give it, yeah.
Very honored to accept the Proctor Medal here at ARVO, and we thank ARVO very much. We're very grateful for being the recipients of the medal. We'd also like to thank both Goldis and Max for their very kind comments. These are our financial disclosures, and this is our All-Star 2 team. These are a fantastic group of collaborators, and we are very grateful for having worked with them for a number of years. Thank you to our funding sources. We've separated them into patient studies as well as laboratory studies. We want to do a very special shout out to the National Eye Institute and the National Institute on Aging. Thank you. Who have funded our research for very many years, and we greatly appreciate that. Here are our takeaway messages for today. Our goal is the prevention of AMD onset, as well as arresting its earliest progression. This is preferable not only to patients, but to families, providers, and payers. It is also very feasible from the standpoint of what's known about cellular level imaging, the accessibility of the eye, the very large biologic effects of the fovea, and also the molecular commonality with atherosclerotic and cardiometabolic disease. Now, rod-mediated dark adaptation, or as we call it, RMDA, because it's such a mouthful, is ideally suited for determining enrollment eligibility for trials in early disease. It also is a potential outcome measure for interventions focused on preventing intermediate AMD, rather than just focusing on trying to preventing the onset of atrophy. RMDA has strong roots in neurophysiology, the progression sequence deposit-driven AMD, the biology of the fovea, and also epidemiology. Delayed RMD also has imaging correlates, which we'll discuss with you today, which really provides insight into the pathophysiology of the early disease. It also can, in terms of imaging outcomes that are functionally validated with RMDA may prove to be the most useful for studying early disease. We also want to mention that natural history studies can inform AMD trials, and we're hoping that All-STAR and All-STAR 2, which I described to you today, will be doing that. They provide information on both incidence and progression. They give us information about potential outcome measures, and they also give us information about patient subpopulations that may be most appropriate for trials. So now I'm going to talk about neurophysiology and psychophysics in terms of the roots of RMDA. So about 25 years ago, I was honored to work with the late Sam Jacobson, and we were both very interested in studying in aging as well as early AMD, scotopic deficits and photopic sensitivity deficits. And we found two things. First of all, let me also mention that in these studies, we used standard AMD classification systems to define our patients, which was very popular in epidemiology at the time, but was not really being used much in psychophysics. So the two things that we found were that first, scotopic deficits and photopic deficits are present in AMD, in early AMD and also in aging. However, what we also found is the scotopic deficits are much more common and tend to be more severe in many patients. So I'd like to also talk about dark adaptation. Dark adaptation is a psychophysical or behavioral test, and it assesses the recovery of light sensitivity after the exposure of a bright light. Now, so dark adaptation has actually been studied for many decades, and actually the initial studies published in many prominent journals occurred in the early part of the 20th century. Now, Lam and Pugh, in a really beautifully written treatise, described the known molecular mechanisms of the retinoid cycle, which is the neurophysiological basis of dark adaptation. It's also important to note that replenishment of 11-cis-retinol to rhodopsin is the rate-limiting step for rod sensitivity recovery in dark adaptation. So we noticed that in an article published in the mid-90s by Alan Bird's group that he looked at dark adaptation in early AMD. And just to get us oriented, this is the cone recovery part, which is not that perturbed in dark adaptation in early AMD. This is the rod recovery part, but in AMD, you'll notice in early AMD, it was delayed. And so basically, he basically demonstrated this phenomenon we became very interested in a small number of patients. Alan Bird's group showed and actually suggested a hypothesis that a localized shortage of vitamin A caused by impair exchange across Bruch's membrane, or possibly progressive metabolic impairment of the RPE, is what is underlying delayed dark adaptation. So we also wanted to establish this in our own laboratory, which we attempted to do in about 2000. We also used, as I mentioned earlier, from our influence in working with Sam Jacobson, a standardized AMD classification system so we could actually more objectively study the presence and the severity of AMD rather than just relying on a single clinician's judgment. And so what we see here on the right with the early AMD patients, delays in dark adaptation, and over on the left, a normal patient where you see dark adaptation is occurring at a much more faster rate. So we were very interested in this suggestion or hypothesis that Bird's group made that basically that the delay in dark adaptation was due to a local shortage of retinol in both aging and early AMD. So we asked the question, what is the impact of a 30-day course of high-dose vitamin A or retinol on the rate of RMDA? So we basically did a randomized experiment where we had half the patients receive a high dose of retinol for 30 days, and the other patients did not receive this. It was double masked, and it was also placebo-controlled. And what we found was very, very interesting. We found that in the retinol group, we could actually speed up dark adaptation. Now, keeping in mind, this was very exciting, but even more so through a patient-reported outcome measure. We showed that those patients who had greater increases in rod slope, that is, their dark adaptation improved the most, on a mobility subscale of the low luminance questionnaire, they reported more improvement. So basically, we were heartened to see that at least our data were consistent with the local shortage of retinol in aging and AMD. So our student and later our colleague, Greg Jackson, actually developed a new RMD metric called rod intercept time, or RIT. This was developed in conjunction with the development of our instrument, the ADAPT-EX. Now, rod intercept time is the duration required for sensitivity to recover to a criterion sensitivity value that is in the latter half of rod-mediated dark adaptation, of rod recovery. And if you look at the plot, you'll see there at log3 sensitivity, if you focus on the blue dots and the red dots over on the left, you'll see that RIT is actually quite low, maybe 8 or 10 minutes. But if you look at the AMD patients over on the right, you'll see it's actually quite delayed, and some people don't even recover within 20 minutes. So we then went about the All-Star study. And now the All-Star study was very interested in patients who were in normal macular health at baseline. And we measured rod-mediated dark adaptation at 5 degrees, or RMD5, as we call it. And what we found is that those people with rod-mediated dark adaptation delays were about 2 times more likely to have incident early AMD 3 years later as compared to those who did not have delayed dark adaptation. This actually showed us that delayed RMD at 5 degrees is really the first functional biomarker of early AMD.
Thank you, Cynthia. My part of this story begins at the microscope. The photoreceptors and RPE together form the sensing chip of the brain, like a smartphone camera, seen here in a flat mount. The fovea in panel A has a long, thin cone inner segments, and the parafovea in B has 3 times larger cone inner segments surrounded by rings of rod inner segments. And the RPE at the same locations in different eyes has geometrically precise skeleton in CD and autofluorescence long-lasting residual bodies in EF. This microscopy is the basis of single-cell imaging of the human retina. These visualization targets helped launch adaptive optics-assisted imaging like this. And this work also provided a census of cells for aging studies as continuous 2-dimensional maps and demonstrated the rod dominance of human retina. The important features are the ring of high rod density on the left and high concentration of foveal cones on the right. The 6-millimeter diameter central area affected by AMD is only 3% of the total retinal area, and it is captured in this clinical ruler called the ETDRS grid. In the next slide, we'll look at a slice across that grid. The y-axis shows the spatial densities of cones, rods, and RPE. In the central 1-millimeter diameter, cone density has a 5-fold gradient. Rods increase from 0 to 35K, and the RPE is almost flat. Rod density goes from 0 to 130K just outside the ETDRS, and the degradative load on the RPE from phagocytosis is thus biphasic. The 3-millimeter diameter macula lutea, or yellow spot, contains the xanthophyll carotenoids lutein and zeaxanthin. The macula lutea is the high-risk area for AMD, as I will elaborate on later. In 1993, we used these novel tissue microscopy and computational methods to count photoreceptors in donor eyes. In AMD, we found preservation of foveal cones on the top and loss of perifoveal rods with misshapen cones on the bottom. In the age series, we found an area of vulnerable rods. The rods are disproportionately lost near the fovea compared to their peak further out. We now test RMDA at 5 degrees because of this. This histology fit with Cynthia's functional testing, showing disproportionate disproportionate loss of rod sensitivity near the fovea than further away. And the cone sensitivity in the same eyes did not show this spatial trend. The model of AMD progression centers around, incorporates the geometry of the fovea and drusen biology, shown on FOSS on the top and a graph of relative harms and protections below. The key elements are transport dysfunction at the level of the choriocapillaris and Brooks membrane, which lead to the high-risk deposits in an oil spill under the fovea. The foveal cones are protected by the macular xanthophyll carotenoids, and these factors are perfectly aligned spatially, and the RMDA test spot is at the rim of this high-risk area. Let us consider aging across the chorioretinal layers for the era of OCT imaging. We compared published tissue-level findings in normal central retinas and ran a line through data at 20 and 90 years. And here is a minimal decline for cones and a 36% decline for rods. The flat line is the RPE. On the right is the choroidal thickness and coverage of Brooks membrane by choriocapillaris. These quantitatively parallel the loss of rods. This suggests that the photoreceptors do poorly because they are far away from the choriocapillaris. In the middle is a 2-fold thickening of Brooks membrane and a remarkable 15-fold increase in histochemically detectable esterified cholesterol. This is a key marker for the lipoprotein pathway of high-risk drusen. Let's look at that 15-fold increase next. This electron micrograph from the Sarks-Killingsworth group in Australia showed what they called membranous debris, the lipid-rich principal component of soft drusen. We showed with Philippine histochemistry that drusen contain unesterified cholesterol called UC, which is consistent with the membranes, but it also showed EC, esterified cholesterol. This is important because the only way to get esterified cholesterol out of a cell is by lipoprotein particles. Brooks membrane markedly filled with esterified cholesterol in normal aging, 7-fold higher in the central area than in the periphery. We showed with lipid-preserving electron microscopy that membranous debris was not membranes but solid particles. And this quick freeze deep etch from Mark Johnson's group showed solid particles with surface and core morphology, just like lipoproteins. We showed mRNA and protein for apolipoprotein B and microsomal triglyceride transfer protein, which together mark the RPE as a constitutive lipoprotein secretor, like the liver and the intestine. And last year, Kathy Bose-Battaglia's lab created an RPE-specific knockout mouse with establishing the lipoprotein pathway as essential to mammalian retinal physiology. Visible drusen in a lump and an invisible basal linear deposit in a thin layer form one continuous oil spill made by functional RPE due to a transport barrier of Brooks membrane and choriocapillaris, and drusen themselves add to the barrier. This is a source of pro-inflammatory intermediates. It's a cleavage plane for invading new vessels, and when drusen get very large, they put the RPE into a state of metabolic insufficiency and hypoxia. And please note this material is found almost exclusively in the central retina. Drusen culminate a process of lipoprotein accumulation throughout adulthood. We use high-resolution histology of many eyes to find that both soft druse material and its precursor pre-basal linear deposit in AMD and control eyes is more common under the fovea than in the perifovea. Is the enormity and spatial specificity of drusen-associated risk evident in population-based epidemiology? Yes, in the few studies that looked. In the Blue Mountain Eye study of Australia provided longitudinal data. The odds ratio for progression to late AMD at 10 years due to drusen at baseline within the ETDRS central subfield is an amazing 26. There was a 3-fold drop-off within the inner ring, that is the macula lutea, and there was no added risk from drusen in the outer ring. And new studies have shown that there is no added risk from drusen way outside the ETDRS. As I mentioned, our histology supports high-risk drusen material in the ETDRS area only, and longitudinal OCT supports the spatial distribution within the ETDRS. Thus, the fovea plus time confers as much progression rich as all the genetic variants, plus lifestyle factors, as shown by the iRisk consortium. Cynthia, this is you. Thank you.
I'd like to talk to you about the All-Star 2 study, which is our second longitudinal study. So before Christine and I got started on this study, we decided to try to develop a theoretical framework for understanding vision progression during the phases of aging to intermediate AMD. And if you start at the bottom, this is aging, and as we move up, it goes up to intermediate AMD. So in aging and in the earliest phases of AMD, what you see are changes in the choriocapillaris, Brooks membrane, RPE, also the emergence of cellular deposits. And what this does is basically, we believe, compromise retinoid resupply, which basically generates a delay in rod-mediated dark adaptation. Oops. As we go back to early AMD in the middle, we see that photoreceptor degeneration starts to occur. And this leads to rod outer segment changes, and even in some cases with early cell death. And we believe this leads to impaired rod-mediated sensitivity losses under steady-state conditions. And then as we move to intermediate AMD, we believe you start to see corrupted neural circuitry. And when this happens, this has a negative impact on pattern vision. So this is where you start to see deficits in acuity and contrast sensitivity, which are measures of spatial vision. So I'd like to talk to you about the design of the All-Star 2 study. It was a longitudinal study with a similar design, at least time-wise, from the original All-Star study, where we had a baseline visit and then a follow-up visit 3 years later. It was a hypothesis-driven observational study where we were trying to understand functionally validated structural endpoints with an emphasis on RMDA5. As you can see here, we enrolled people aged 60 years and over from comprehensive eye care practices at UAB. We also used the AREDS 9-step system to classify into 3 groups based on their fundus photos: normal macular health, early AMD, and intermediate AMD. We also tested very many visual functions in addition to RMDA because we thought it was very important during the study to compare all these measures that everybody was studying in the literature, including acuity, contrast sensitivity, light sensitivity under different luminance conditions. So they were all included. We also had a very busy imaging protocol as well, with all these types of imaging modalities done in All-Star 2. We're publishing this data now, it's coming out, but many of these presentations are also being presented here at ARVO this week. So you can see all these visual functions on the left, but what we wanted to do is we compared them, and what we found is that all the visual functions, every one of them that we studied, were worse in intermediate AMD compared to what was happening in normal macular health. But the interesting thing is that the only visual function worse in early AMD as compared to normal is rod-mediated dark adaptation. Again, cross-sectionally underscoring that this was the first functional biomarker. We also then took all our visual functions and we converted them to z-scores because we wanted to compare among them because they were all in different types of units. And basically what we found is that RMD5 has the largest range of z-scores from normal to intermediate AMD. And you can see it's in sharp contrast to the other visual functions. For example, other tests were either half or less than half, had half of this range. Even, for example, scotopic sensitivity. And as you can see, tests of photopic vision are the least discriminatory. We also were interested in whether RMD5 was repeatable. And the reason we were interested in this, if you're going to use it in a trial, as you know, you have to have a repeatable measurement. And we looked at it in normal as well as in early and intermediate AMD. And what we found is that the intra-class correlation coefficient was in fact very high. So the answer is yes, it is repeatable. We were also interested in cataract. As we all know, cataract is very, very common in older adults. Some people have intraocular lenses, some don't. But in terms of enrollment in studies and enrollment in trials, we were curious as to what level of cataract might be appropriate, if any. And what we found is that if we compared people before and after cataract surgery, and we looked at the level of their cataract at baseline, and we found that in terms of the lens classification system called LOCS-3, if the score was less than 4, as you can see there on the top, the RMDA5 was highly repeatable. However, if the LOCS-3 score was greater than or equal to 4, you can actually see post-cataract surgery, there was a bigger difference. And so basically, after cataract surgery with cataracts of this level, you see a difference on average about 3 to 6 minutes in terms of RIT. Now, we were also interested in All-Star 2 in terms of the incidence of AMD. Of course, this looks at the rate in which new cases occur in 3 years. Now, we had a number over 200 normal eyes at baseline. Over 3 years, 26 of these eyes converted to AMD in 3 years. And what we found is that slow RMDA eyes were about 3.5 times more likely to have AMD at follow-up. But look at the other visual functions. If these are confidence intervals, nothing compares to RMDA5, and they were not statistically significant with the possible exception of low luminance visual acuity, which was weaker. Now, we were also interested in the progression of AMD. So here, we're focusing at baseline on over 100 eyes who had early AMD at baseline. 34 of these early AMD eyes progressed to worse AMD in 3 years. And also, what we found was that slow RMDA were almost 4 times more likely to progress. But again, look at the other visual functions. They're really not associated with progression. Christine.
Thank you. So in All-Star 2, we're benchmarking the imaging tests with RMDA5. And we aim to develop snapshot assessments that are quicker than RMDA, as well as develop a cellular level progression sequence. And these include hypothesis testing and explorations of the neural and vascular origins of AMD. The interdigitation zone, or the IZ of spectral domain OCT, includes the RPE apical processes with melanosomes in them, as shown for this one reconstructed cell. This molecularly distinct domain has junctions and transporters, and importantly, retinoid binding proteins. In a pilot study, we compared young and aged normal adults and found that the visibility of this band decreases in the aged eyes. Our collaborators, Vas Sada and Jewel Hu, used the OCT volumes from the All-Star 2 baseline visit and found that the area of preserved IZ is much smaller than the area of preserved ellipsoid zone, or EZ, which represents the photoreceptor inner segments. The intact area was strongly correlated with RMDA5, more so than the EZ. Does analysis of OCT angiography of chorea capillaris support a transport dysfunction, as predicted by our hypothesis? Using spectral domain OCTA, we determined the percentage of flow deficit, which appears as dark pixels in these images. We used 2 adjacent scans to check the fovea and the superior retina. In these maps of 3 diagnostic groups, we see the flow signal deficit shown here as warm colors is worse under the fovea in intermediate AMD, but also to a lesser extent in normal and early AMD. And here is the RMDA5 test location. So OCTA signal is worse under the fovea, even in eyes with delayed RMDA5. These maps show Spearman structure-function correlations between flow signal deficit and RMDA, with RMDA5 at the gray spot. The flow signal deficits under the fovea were strongly associated with slow RMDA. Compared to RMDA5, the correlation of cone-mediated acuity with flow deficit was much lower, 0.21 versus 0.52. So even though the flow signal is worse at the fovea, rod vision suffers more than cone vision. Let us address the question of whether subretinal drusenoid deposit, or SDD, also called reticular pseudodrusen, is a factor, and we feel it's an indicator of dysfunctional transport. Spectral domain OCT revealed reflective material between the RPE and the photoreceptors, leading to this Zweifel scale. Our histology confirmed material which was seen by the SARCs, but not named, in this location. We found that SDD was thick where rods are numerous, and the drusen material was thick where the cones were numerous. So we proposed that the deposits represented dysregulation of lipid transfers, serving cone and rod outer segment membrane maintenance. In 2015, Kathy Kukris and the National Eye Institute group showed that eyes with reticular pseudodrusen had an extremely delayed RMDA, shown here by that blue line. So is this the case for RMDA5 in the All-Star 2 participants? Yes. In early and intermediate AMD, rod intercept time is longer when SDD is present, yet even in eyes, in intermediate AMD eyes without SDD, RMDA is delayed. Okay, does SDD really track rods? That's an important question. But yes, but other factors are important. In All-Star 2, the extent of SDD is moderately and quite specifically correlated with RMDA5. Well, how do we measure the extent of SDD? We applied deep learning to 55-degree near-infrared reflectance images, which are over 6 times the size of the ETDRS grid, because we wanted to see if we could capture the entire ring of high rod density. And we found, like many others, that SDD are abundant in the superior outer ETDRS, but we also found that the SDD peak is closer to the fovea than the peak of the rod density. And in fact, the RMDA5 test target location, which is chosen many years ago, it precisely probes this point. In aged eyes, we found that SDD appears first next to the optic nerve head, as we saw in histology. Early AMD has a light sprinkling throughout this central area, and intermediate AMD has most of the SDD in these persons. So yes, SDD appears where there are many rods, but the distribution also reveals vascular origins and proximity to the fovea as important factors. Fluorescence lifetime imaging ophthalmoscopy, or FLIO, is a metabolic imaging technique that records the time that a fluorophore stays in an activated state. A seminal study by Sauer and Bernstein showed that a third of age-normal eyes and all AMD eyes had lengthened lifetimes in the long spectral channel, especially in the ETDS or outer ring. So we hypothesized that the longer lifetimes would correlate with delayed RMDA5 and possibly represent the beginnings of SDD. And indeed, we found that FLIO long spectral channel correlated strongly with RIT, especially in the outer ETDRS. And these correlations are the strongest so far. FLIO was uncorrelated with photopic vision, and furthermore, we interestingly saw, like Sauer and Bernstein, that the lifetimes are much worse near the optic nerve head, as shown in these new meridian plots. And recall that the choroid is thin between the fovea and the optic nerve at all ages, as shown by the Rosenfeld-Wong team. So I'm going to assemble a hypothesis for deposit-driven AMD as heralded by delayed RMDA5. I'm going to link it to the ARMS2 HEDER-A1 locus, which is the largest single genetic association. So let's propose that there are 2 dials to control deposit formation. One is a constitutive release of material from outer retinal cells, and the other is a flow control of this output to the circulation. And why is this massive outflow? I would propose that it's to dispose of materials either generated by or left over from diet. And the deposits form where the cells have high metabolic need, like the fovea, or transport to the circulation is impaired. So the slide shows a sequential barrier of AMD deposits. On the left are 5 differential planes, differential dysregulated outflow starting at the choriocapillaris endothelium across 5 layers moving inward, starting with lipoproteins and Brooks membrane, soft drusen precursor, the oil spill itself, other trapped material, and finally, the RPE itself is the barrier when the SDD appear. And we propose that this whole thing starts at the fovea and gradually spreads outward to the rod-containing areas, so that delayed RMDA5, which starts in aging, indicates dysfunction long before any of this pathology shows up. And this provides new impetus to explore the ARMS2 association with delayed RMDA5 in normal eyes, which we reported in 2019 for the first All-Star. And the same eyes showed no association with complement factor H. And in a manuscript under review, we showed that this association shows for RMDA5 only, and that's the only visual function. And this converges very nicely with recent data from Roy Schwartz and the Moorfields group, showing a clear association between ARMS2 HEDER-A1 locus and high SDD load in eyes lacking visible drusen. So the future for RMDA5 is varied. We wish to know what RMDA5 means for tasks performed in dim light, like reading, like mobility. We'd like to know if these visibility problems are associated with patient-reported outcomes, like a low luminance questionnaire that could be used in a trial. And finally, we wish to shorten the test. Note the second slope of the RMDA curve is constant over many light levels. On the precision prevention side, we are building tools to monitor foveal nutrition. The macular xanthophyll carotenoids, lutein zeaxanthin, localized to foveal neurons and Mueller glia, and their bioavailability is regulated by the HDL pathway of AMD genetics. And with new cellular level hypotheses, we're developing imaging metrics for 2-wavelength autofluorescence, which is shown on the right. So to returning to the takeaway, our goal is precision prevention of AMD onset and early progression, way before neovascularization or atrophy. RMDA has strong roots in neurophysiology, AMD progression, neurobiology of the fovea, and epidemiology, and it's well suited for determining enrollment, eligibility for trials in early disease, and promising imaging correlates to go along. I'd like to thank you for your attention, and I'd like to thank all of you for your collaboration and friendship over 40 years. Thank you.